In this screencast, we're going to be talking about Biozone worksheet number 52, investigating catalase activity. It's important that you're able to talk about how you can investigate the rate of an enzymatic reaction in a number of different scenarios. In this particular scenario, the substrate involved is hydrogen peroxide. Hydrogen peroxide it's something you can buy at a pharmacy. It kind of bubbles. It often you use it to clean cuts and other things. And it's essentially, two hydrogens and two oxygens. And organisms and cells make hydrogen peroxide as a byproduct of a lot of metabolic activity. And that hydrogen peroxide will actually decompose on its own into water and oxygen gas. So the reactant or the substrate is hydrogen peroxide, and the products are water and oxygen gas. Cells will produce hydrogen peroxide as a byproduct of metabolism, but they want to get rid of the hydrogen peroxide because it can be corrosive in cells. It causes a lot of oxidation. And so most organisms make an enzyme that can break down hydrogen peroxide. The mung beans, which are the organism we're going to be using to create the enzyme, or that the enzyme's already in, they make an enzyme called catalase. There's also another enzyme that we'll use later on when we're actually carrying out a lab, is peroxidase. Okay, so here's the substrate, hydrogen peroxide. And here is essentially the mung beans that have the enzyme in it, the catalase. So when they put the hydrogen peroxide into this mung bean slurry, these 10 grams of mung beans, that's going to produce a bunch of oxygen gas, right, and water, because the hydrogen peroxide will break down in the presence of catalase into water and oxygen gas. Well, the oxygen gas, because it takes up more volume than a liquid would, is going to move down this tube and then displace the water that's in this little apparatus. There's a cylinder turned upside down. So as more and more oxygen bubbles go here, you'll see the water moving down along this graduated cylinder, so you can actually record how much oxygen is produced in a certain amount of time. So the dependent variable in this lab is essentially going to be the rate of the decomposition of hydrogen peroxide, right, into water and oxygen gas. Okay, so how quickly is that happening? We'll eventually have it be in centimeters per second per gram of mung bean. Okay, and then the independent variable in this lab is they take mung beans that have been germinating at different stages. So germinating is when seeds break their dormancy and start to slowly grow into plants. And so they're really metabolically active. So the more metabolically active they are, it's likely that they'll be producing more of the catalase enzyme to break down hydrogen peroxide. And so as the seeds are super metabolically active, there's probably a point in their germination stage that they're the most metabolically active, and that's what we're going to find out, is how many days of germination, if we test these seeds at different stages of germination, how much catalase do they have, essentially, So the independent variable is the stage of germination, and of course the dependent variable then would be the enzyme activity of that catalase, or the decomposition of hydrogen peroxide into oxygen. But we'll be measuring the amount of oxygen gas that's been created. Okay, in the next screencast, we're going to be looking at how to actually use Excel to be able to graph that.